In the name of Allah, the Beneficent, the Merciful. Sequencing can be used to identify types of mutations. Sequencing involves determination of the base sequence of DNA. In this method, the single-stranded DNA to be sequenced is used as a template for DNA synthesis by DNA polymerase. A radiolabeled primer complementary to 3' end of target DNA is added along with 4 deoxynucleoside triphosphates. The sample is divided into 4 reaction tubes and a small amount of one of the 4 dideoxynucleoside triphosphates linked to fluorescent dyes are added to each tube. In the first reaction tube, adenosine dideoxynucleotide triphosphate ddATP, which lacks 2' and 3' hydroxyl groups, is added in little amount. The ddATP can be inserted into the growing chain of DNA, but after its insertion, further elongation cannot take place and elongation is stopped. As a result, different sized DNA fragments are produced. In the second reaction tube, cytidine dideoxynucleotide triphosphate ddCTP lacking 3' and 2' hydroxyl groups is added in a little amount. The ddCTP can be inserted into the growing chain of DNA, but after its insertion, further elongation cannot take place and elongation is stopped. As a result, the DNA fragments of varying lengths are produced. In third reaction tube, guanosine dideoxynucleotide triphosphate ddGTP deficient in 3' and 2' hydroxyl groups is added in little amount. The ddGTP can be inserted into the growing chain of the DNA. So that's the ddGTP, and it can be inserted into the growing chain of the DNA, but after its insertion, elongation is stopped. As a result, DNA fragments of varying lengths are produced. In fourth reaction tube, thymidine dideoxynucleotide triphosphate ddTTP is added in little amount. The ddTTP can be inserted into the growing chain of the DNA. So that's the ddTTP. It lacks the 3' and 2' hydroxyl groups, and that is the elongating chain, and as the ddTTP is inserted, elongation is stopped. As a result, DNA fragments of varying lengths are produced. The pieces of newly synthesized DNA in each tube are separated by capillary electrophoresis in a different lane of the gel. The sequence of new strand can be read from the smallest to the largest fragments on the gel, for example, from bottom to the top, or from 5' towards the 3' end. So reading the sequence from bottom to top makes it C T T G G A A C T G T A. When C corresponds to the 10 nucleotide fragment, T corresponds to 11 nucleotide fragment, T at end of 12 nucleotide fragment, G at the end of 13 and 14 nucleotide fragments consecutively, followed by adenines at ends of 15 and 16 nucleotide fragments, cytosine at the end of 17 nucleotide fragment, thymine at the end of 18 nucleotide fragment, G again at the end of 19 nucleotide fragment, T at the end of 20 nucleotide fragment, and A at the end of 21st nucleotide fragment. Normally, the 3' hydroxyl group of deoxyribose of one nucleotide forms a bond with the 5' hydroxyl group of an adjacent nucleotide through a phosphoryl group. In ddnucleotide triphosphates, both 3 and 5', both 2 and 3' ends are unavailable. Therefore, the chain terminates when these are inserted. The main limitation of Sanger dideoxy sequencing method is speed. Improvements in this technique have led to next generation sequencing, which allows the whole genome to be sequenced in one day. It involves mechanical fractionation of genome followed by rapid sequencing and alignment. Many segments are sequenced simultaneously. The sequences are read from bottom to top of trail. For normal, or control, it makes AA, T, A, T, C, A, T, C, triple T, double G, T. For cystic fibrosis, it can be read as AA, T, A, T, C, A, T, T, G, G, T, G, T, T. In this case, the sequence for the control and cystic fibrosis are identical for the first 8 bases. Positions of 15 to 12 of normal gene and 9 to 12 of cystic fibrosis gene are identical. Therefore, there is a 3 base deletion in cystic fibrosis gene corresponding to the 9 to 11 of normal gene. Since it is a deletion of 3 bases, therefore some researchers do not classify it as a frame shift mutation. The DNA pattern is indicative of the deletion of codon leading to absence of phenylalanine at 508th position in the CFTR. So the sample of DNA to be sequenced is added to a mixture which contains the single-stranded DNA which is to be sequenced along with the DNA polymerase and deoxyribonucleotide triphosphates of the 4 types. Then this mixture is divided into 4 tubes, and in each tube a ddNTP is added. The ddNTP will be inserted into the growing chain, and it will lead to the formation of different sized fragments which are separated by gel electrophoresis, and the sequence can be read from the bottom towards the top.